Hi there, in this video we are going to start lipid biochemistry. So lipids are basically complex compound made up of fatty acids. So what are fatty acids? Fatty acids are basically carboxylic acids, or we define them as long chain carboxylic acids. However, they can be short chains. The general formula of fatty acid is R into CH2 COOH, or you can write it as CH3 CH2N COOH. So this is the general formula of fatty acid, where here is N and N can be any number. So this portion of fatty acid is hydrophobic. This is the hydrophobic portion, or it flies away from the water, and this portion is hydrophilic. It is polar and it is hydrophilic. So fatty acids are amphipathic compounds. They have both polar and non-polar regions. However, if the hydrophobic region increases, if the chain length increases, as chain length increases, the hydrophobic portion becomes dominant. So in the long chain fatty acids having very long hydrophobic tails or chains, they don't dissolve in water and blood, and they need to be transported in association with plasma proteins, for example albumin, plasma proteins, for example albumin, or lipoproteins. So fatty acids are carboxylic acid. We define them as long chain carboxylic acid. General formula is CH3 CH2N in COOH. This portion is hydrophobic, this portion is hydrophilic. That's why they are amphipathic compound, and when their chain length increases, their hydrophobic nature becomes dominant. So long chain fatty acid require plasma protein, for example albumin, or lipoproteins, which are amphipathic compound, to dissolve them in blood and to be transported in blood. So this is the primarily structure of fatty acid, and fatty acid can be saturated as well as unsaturated. So unsaturated fatty acid have low melting point, and fatty acid having long chain fatty acids have high melting point. As chain length increases, melting point increases. As unsaturation increases, melting point decreases. And the fatty acid, saturated fatty acid are usually straight. However, unsaturated fatty acid have a nick or bend in their tails, as we show for phospholipid. This is saturated phospholipid having straight tails. However, unsaturated phospholipid have a bend or nick in its tail. So this is the concept of saturation. Now we come towards the numbering of fatty acid. For example, we take an example of linoleic acid. Linoleic acid here, and linoleic acid is written as 18:2 and then 9:12. What does it mean? It means that linoleic acid has 18 number of carbon atoms. It indicates number of carbon atoms, so it has 18 number of carbon atoms. This 2 after the colon indicates that it has 2 double bonds. It shows the number of double bonds. At this 9:12 shows the location of double bond. So first double bond is between 9 and 10 carbon atoms, and second double bond is between 12 and 13 carbon atoms. We take another example here, arachidonic acid. So it is written as 20:4 and 5, 8, 11, 14. So 20 indicates the number of carbon atom. It has the 20 number of carbon atoms, and here 4 indicates number of double bonds. It has 4 double bonds, and here is the location of double bond. First double bond between 5 and 6 carbon, then 8 and 9 carbon, then 11 and 12 carbon, then 14 and 15th carbon atom. So in the structure of fatty acid, for example, this is the structure of fatty acid. The carboxylic carbon is always written as 1. This is the first carbon, it is C1, it is C2, it is C3, and it is C4, and this carbon is also called alpha carbon. So the carbon that has functional group is called alpha carbon. So this carbon is having functional group, so it is alpha carbon. The next to it is beta carbon, next to it is gamma carbon. And if the chain continues, the end carbon is termed as omega carbon. We can count the double bond from omega end as well. For example, if in arachidonic acid we count the double bond from other end, then from other end this 14 comes about 7th carbon atom, and 15 come about 6th carbon atom. So it comes about omega 6 fatty acid as well. because from its 20th carbon atom, arachidonic acid has 20 carbons. So its last double bond is between C14 and 15. So from 20th, if we count 20, 19, 18, 17 and 16 and then 15. So it comes about 1, 2, 3, 4, 5, 6. So it is omega 6 fatty acid because it's from omega end, from other end or omega end, its first double bond is at 6th position. So it is an omega 6 fatty acid. Similarly, if it comes towards linoleic acid, so it has 18 carbon atom and its double bond is between 12 and 13. If we count from omega end, then it comes about C18, C17, C16, C15, C14 and C13. So it comes about 1, 2, 3, 4, 5, 6. So it is also omega 6 fatty acid. So this is the concept of omega fatty acid, in which we count double bonds from the omega end of the fatty acid. So two fatty acids are very important: linoleic acid and alpha linolenic acid. Linoleic acid is 18:2 and 9:12, and alpha linolenic acid is 18:3, 9, 12 and 15. So these both are essential fatty acids. We cannot synthesize them. So if diet is deficient, it leads to diarrhea, dermatitis and dementia. So these are the symptoms of deficiency of essential fatty acids, and we obtain them from plants. And linolenic acid also synthesizes arachidonic acid. So if linolenic acid is deficient, arachidonic acid also become deficient or essential. So these fatty acids are essential, we cannot synthesize them, and they must be supplied by diet, and they usually come from the plant-based diet. Okay. Next we come towards the two concept, the concept of ketone bodies. So what are ketone bodies? Uh, Ketone bodies, basically three substances are termed as ketone bodies. One is acetone, other one is acetate and beta hydroxy butyrate. These three compounds are called ketone bodies, and they are produced from the acetyl CoA. They are produced from acetyl CoA, and this acetyl CoA is CH3, C double bond with oxygen, then S and then CoA. It is produced from the fatty acid oxidation. It is produced from fatty acid oxidation. So when body breaks down lipids inside the body are broken down into the fatty acids, and these fatty acids are broken down into the acetyl CoA. This acetyl CoA goes to the liver and enters into the tricarboxylic acid cycle or Krebs cycle, and where it is converted to NADH, FADH2 and GTP, which yields energy in the form of ATPs on the electron transport chain. So ketone bodies are produced from this acetyl CoA. Let's discuss. Lipids are broken down to fatty acids. Fatty acids are converted to acetyl CoA. Acetyl CoA goes to liver. Liver oxidizes them by tricarboxylic acid cycle or Krebs cycle and converts them to NADH, FADH2 and GTP, which are used to produce ATP by electron transport chain. However, there is a limit to which liver can oxidize acetyl CoA. If the production of acetyl CoA increases too much due to breakdown of lipid, if breakdown of lipid increases and acetyl CoA production increases, then what happens? The capacity of liver to oxidize them by TSA cycle is accomplished. Now the additional acetyl CoA is diverted to the production of ketone bodies. And why this lipid breakdown can increase? It can increase in fasting, when a person is fasting, and it can increase in diabetes. It can increase in diabetes, usually diabetes type 1. So when the production of acetyl CoA has increased, liver converts them to ketone bodies. So these ketone bodies are polar compounds. They dissolve in blood. They don't need any transporter protein to be transported in blood, and they are used by peripheral tissues as an additional fuel. Muscles can use them, nerves can use them, brain can use them. So they spare the glucose. So they have glucose sparing effect on the body. So if there is some defect in the lipid metabolism, acetyl CoA is not produced. When acetyl CoA is not produced, ketone body is not produced. So there will be hypoketosis. When there is defect in lipid metabolism, there will be hypoketosis. Moreover, when there are no ketone bodies, then reliance on glucose increases or glucose consumption increases, so it can lead to the hypoglycemia. So defect of lipid metabolism can lead to the hypoketosis as well as hypoglycemia, because ketone bodies production is decreased and reliance on glucose increased. So why diabetes causes increased production of ketone bodies? In diabetes, insulin is low, and glucagon is increased. So increased glucagon leads to the lipolysis. So this lipolysis yields fatty acids. These fatty acids converted to acetyl CoA, and acetyl CoA is converted to ketone bodies. So it can lead to the ketoacidosis. So in type 1 diabetes, due to decreased insulin and increased glucagon, lipolysis occurs, which yields fatty acid, and these fatty acids converted to acetyl CoA, which lead to the ketoacidosis or diabetic ketoacidosis, which is termed as DKA. What happens in this condition? These three compounds increase in the body. They release H positive in the blood, due to which blood pH falls, which is called acidosis, and moreover they also release in urine, which leads to the ketonuria, and they are osmotically active, they withdraw water with them, so it leads to the dehydration as well. So what happens in diabetes? Due to increased production of ketone bodies, there is decline in pH, which is acidosis, and there is dehydration as well due to loss of water. So it leads to the diabetic ketoacidosis, which is the feature of type 1 diabetes. Type 1 diabetes, it does not occur usually in type 2, because in type 2 insulin, some amount of insulin is present. Insulin present in type 2, there is insulin resistance. However, the presence of insulin prevents the production of ketone bodies and ketoacidosis to occur. So it is the case which happens in the type 1 diabetes, and the patient suffering from diabetic ketoacidosis yields fruity smell because of the acetone, because acetone has a fruity smell. So it was the portion from the chapter 16 of Lippincott. Next we come toward the chapter 17. So in chapter 17 there are few topics which need to be done. We should be knowing first the structure of glycerol, CH2OH, CHOH, CH2OH. So this is the structure of glycerol, and their phospholipids. Phospholipids are basically of two kinds, which we will be discussing here. Phospholipids can be glycerophospholipids, glycerophospholipids, and sphingophospholipids, sphingophospholipid. So glycerophospholipids have this glycerol as their backbone. This is glycerol, and when this glycerol reacts with fatty acids, let's say the fatty acid C double bond oxygen OH, and then here we write CH2, or you can write R here simply, R, and OH, C double bond oxygen R, and here is phosphate, third carbon, leave it. So it gets converted to CH2. Here it reacts, here water molecule released, so it gets converted to CH2O, C double bond oxygen R, this is C1, add C2, CH O, C double bond oxygen R, and at C3 the compound that joins at C3 is phosphate. So this structure formed is called phosphatidic acid. Phosphatidic acid, you should be knowing its basic structure. This is glycerol and this is phosphatidic acid, and this bond is ester bond. Ester bond is C double bond oxygen O C C, or you can write R here. So this is the ester bond. So fatty acid has joined glycerol by ester bond. This is phosphatidic acid, which is the backbone of phospholipids, glycerophospholipids. This phosphatidic acid joins with many bases to produce other phospholipids. For example, this phosphatidic acid joins with serine, which is a base, and converts to phosphatidyl serine, which is a phospholipid. Phosphatidic acid joins with choline and gets converted to phosphatidyl choline, which is also a phospholipid. It joins ethanolamine and gets converted to phosphatidyl ethanolamine, which is also a phospholipid. It joins with inositol and gets converted to phosphatidyl inositol, which is also a phospholipid. So in this way it gets converted to different kinds of phospholipids. Another phospholipid we should be knowing here is cardiolipin. When two molecules of phosphatidic acid are joined together by glycerol, then the structure formed is called cardiolipin, or you can say if this glycerol here we join a phosphatidic acid and here we join a phosphatidic acid, then the structure form is called cardiolipin. It is found in both prokaryotes as well as eukaryotes. In prokaryotes, for example, this is bacteria here, it is found in this membrane and act as an antigen. This antigen is detected by body immune system and produce antibody against it. So it provides antigenicity to prokaryotes. However, in eukaryotes, it is found in the inner mitochondrial membrane. In eukaryotes, it is found in the mitochondrial membranes, and in mitochondrial membranes it maintains the respiratory complexes which are cytochromes which are involved in electron transport chain. So in the mitochondrial membrane it maintains respiratory complexes. This is the role of cardiolipin uh, in cardiolipin. And then we come towards plasmalogens. Okay, so these are also phospholipids. These phospholipids are produced when fatty acid, we previously discussed that in glycerol this is here join the phosphate, okay, and here at these two compounds are fatty acid joined by ester linkage, O C double bond oxygen R, O C double bond oxygen R. Okay, so if at C1 we replace this fatty acid by an unsaturated alkene, and its chain continues, then the structure formed is called plasmalogen, and the bond form here is ether linkage. This is because this is a COC linkage or ROR linkage, which is an ether linkage. So when an alkene by ether linkage joins C1, then the structure form is plasmalogen. This plasmalogen, the example here given are two, and if here with the phosphate we assign base ethanolamine, then it becomes phosphatidyl ethanolamine, and if here we attach choline, then structure becomes phosphatidyl choline. It is different from the previously formed phospholipid in which phosphatidic acid joins with choline, then structure form is phosphatidyl choline. However, here structure form is phosphatidyl ethanolamine, and previously formed was structure was phosphatidic acid plus ethanolamine was phosphatidic acid phosphatidyl ethanolamine. So when base combined with phosphatidic acid, then DYL used. When ether linkage here is present with alkene, then DAL is used. So these are plasmalogen. They are basically play their role in the regulation of membrane. They maintain membranes. And moreover they are antioxidants, so they prevent from the free radical injury. Example here is phosphatidyl ethanolamine, which is found in nerve tissues and maintain the nerve integrity. Their decreasing level is linked to neurodegenerative disorders. For example, Alzheimer's disease. Phosphatidyl choline is found in the heart and maintains the cardiac health. So next we should be knowing another factor, which is platelet activating factor. Platelet activating factor activates platelets during the acute inflammatory response or cellular injury. So if here the structure replaced is saturated compound, if here by ether linkage a saturated compound is attached, then and here an acetyl group is attached at C2, and acetyl group is attached. Acetyl group is O C double bond oxygen CH3. Then the structure formed is platelet activating factor. This platelet activating factor, here we can attach base, for example choline. So this platelet activating factor is involved in inflammation, and it activates WBCs, and it is highly potent molecule. It acts at low concentration as 10 raised to power minus 12 mole per liter. So these were the different glycerophospholipid. We next come towards sphingophospholipid. We should be knowing sphingosine. Sphingosine is the base, as we write here CH2NH2 and then CH2OH, and here the structure is CH double bond CH, then CH2 12 CH3. This is the base which is called sphingosine. It is involved in structure of sphingomyelin. So when this NH group is attached to some fatty acid. It is attached to some fatty acid. Then this structure formed is called ceramide. This structure then formed is called ceramide. So when sphingosine join with the fatty acid at C2, structure formed is called ceramide, and when the ceramide attach at C1 with phosphorylcholine, P for phosphorus, and then choline. It joins choline here. Then the structure formed is called sphingomyelin. Sphingomyelin is only the sphingolipid found in human, and it is involved in the myelination of nerve fibers. So this is sphingophospholipid, which is sphingomyelin, which is backbone formed is by sphingosine, and sphingosine joined with uh, fatty acid at C2, and at C1 it is attached with phosphorylcholine to form sphingomyelin, which is formed with myelin sheath. So another, we should be knowing the role of phosphatidylinositol, the glycerophospholipid here. Phosphatidylinositol is found in the cellular membrane. It is involved in the GQ signaling pathway, in which it was found in the form of PIP2 phosphatidylinositol bisphosphate that was being cleaved to diacylglycerol and inositol 5-phosphate. We need not to go into detail. Another function we should be knowing is the membrane holding function. It is found in the RBCs, and the RBC's membrane, it is glycosylated, it is attached to carbohydrates, so we call it glycosylated phosphatidylinositol. So it here anchors a protein which is called decay accelerating factor. This decay accelerating factor cleaves complement proteins. We know that there are certain complement proteins in the body, which include C3, C4, C5 and lytic complex and so on, which lead to the lysis of the cells. So decay accelerating factor here present on the surface of RBCs anchor to glycosylated phosphatidylinositol cleaves complement protein. However, when there is some defect in phosphatidylinositol, this anchoring is not present, decay accelerating factor is not present, complement protein become active and lead to the lysis of the RBCs. And when RBCs are lysed, there is anemia, and it is commonly seen in a disease, paroxysmal nocturnal hemoglobinuria. What happens in this disease, paroxysmal nocturnal hemoglobinuria? This linkage GPI is absent, so decay accelerating factor is absent. There is lysis of RBCs, and this lysis usually happens at night. It usually happens at night because at night there is shallow breathing. We breathe shallow at night, so a patient breathes shallow. As a result, H2CO3 accumulates in the body, due to which pH falls. This decrease in pH activates complement and lead to the lysis of RBC. Okay, so this is the principal role of phosphatidylinositol. Another role of phosphatidylinositol is it holds the phospho it holds acetylcholinesterase at the postsynaptic membrane. It holds acetylcholinesterase at postsynaptic membrane and plays its role in the breakdown of acetylcholine. Moreover, it is also holds the phosphatase, alkaline phosphatase, as the intestinal brush border enzyme, and played its role in the digestion. So it was all about today's lecture about lipids. Thank you.